prepare a site for which we will have our fish ponds. Okay, so we have done all the, we have looked at all the criteria selection and everything. And then we want to prepare our fish before we prepare the pond before we receive the fish. Okay, so so we did drying of the pond and then liming and fertilization of the water, right? So this is just a continuation of that lecture, but it is also kind of like we have already discussed about grouper culture. So more or less just strengthening that knowledge and information from all these lectures that we have learned, right? So this is kind of like just summarizing or going through all of the things that I've mentioned before. Okay. Um, so these are the things that, wait, hold on, 2 minutes. Give me 2 minutes, right? Um, Hey guys, so sorry. I just need to sort this camera thing. I'm not used to this. Right, I just see if it works. All right, it's all right now. Okay, let's move on. All right, after we prepare the pond, so there are a few things that we need to keep in mind. Um, these are the things that we need to consider once we have the fish in our farm, okay? So firstly is handling live fish, monitoring and keeping a good record. Think about fish feeding and the feed that we are going to use. And after all of the culturing has been done, we need to think about harvesting, all right, and therefore transporting for the Asian Southeast Asian economy or business restaurants. Usually we would want a feed, live fish, and then so if it is not live, so certain fish needs to be processed, marketed. All right. All right, handling live fish on the farm. Basically, what I'm talking about here is monitoring of fish growth and health. So what are the equipments that will be used? And usually it is a majority of it about maintaining or monitoring the water quality so that the growing and the health of the fish is optimized. Another thing that is very important is taking measurement of length of the fish, and also usually when you take length in fish, it somehow correlates to the weight, but not necessarily. So this is done so that we are able to monitor the feed intake and also the stress signs. All right, usually when fish are sick, they shy away from feeding. So that is one of the first sign that we can look into, observe if the fish are not well, right? And if the water quality is not good, right? So in fish health, it is always about prevention, because once disease has come into your farm or into the farm, it is very hard to treat, and the control measures or the losses are quite high. Therefore, in order to prevent diseases, we need to think about the health of the fish, think about the environment stresses. All right, I've talked about this before. There's 3 stresses, all right, physical, chemical, and perceived stresses in fish, and also virulent pathogens. Right, fish are constantly being bathed, or they are being exposed to the pathogens around them constantly, but what will tip the balance is your water quality, your stresses, and also the health of the host, right? Nutritionally, especially. Okay. I think I've shown this before. It is just us going into a farm and then taking, using a water meter, right? Water quality meter. So this is a multiple parameters. And this is a basic kit. We have they call it a master freshwater test kit. So this basically shows what ammonia. So you can advise the farmers to have this, if not the expensive meter, you can have a test kit that you can use for a few hundred tests. So one master kit is around 120 ringgit. Okay, so one test kit, it's like a big box, comes in a box. So, have that, can advise them, if they do not have even the most basic things, right? The first thing that you would want to suggest is to get a water test kit, a very basic one, all right? Keep it in the farm, and if there are anything that is, concerning, test the water first, all right? I will say this to students: always test the water first. Water is usually is the culprit behind stress in fish. All right, so yeah, this is a picture. So it comes in a very convenient box. This is freshwater. Yeah, this is freshwater. There's also a marine one, which is slightly different. So it says here it can test over for 800 tests, right? So it can test for pH, all right, a higher range of pH, ammonia, nitrite, and nitrate. All right, so usually our concern is with ammonia. Okay, even the slightest, all right, even 0.5, 0.25, depending on the fish species. Slight elevation will cause, like suffocation to the fish, all right? As well as nitrate, not so. Nitrate is nitrate, right? So I'm talking about nitrate here. Nitrate is usually associated with ponds that did not change their water. So usually they would change their water every, usually 30% on a daily basis. Some RAS system, they change every day, right? Regularly 300% daily. So those kind of like stagnant pond do not have a good circulation or water outlet in and out. So the problem is usually with nitrate. So if there are the biological filtration is not good, right? No, the good bacteria will not be able to survive, then you would be concerned about ammonia and nitrate lah. All right. So those are kind of like the basic things that you can look into. Okay, for our unit, aquatic animal health unit, we have 3 sets of this, right? Not this exact meter, but it's the same brand, YSI. So it comes in this like a handheld meter, right? So it's very convenient. You can hold it in with, like that. And then, so this is the probe, right? So you dip the probe in. So if you want to take a measurement, so you want to take a few areas lah in the same pond, but they also have like this cap lah that you can put into. Sorry, this cap is to make sure that the probe is clean. Okay. All right. This is another thing. To ensure that your fish are taking the feed well, they are eating, they're healthy. All right. Some usually they do it not every day, maybe once a week, right? To take measurement of fish. Okay. Traditionally, we have this kind of like either wooden or metal measuring lah kan, measuring apa tu? Measuring ruler or whatever you call it. okay, measuring board or something. But I've always used app. I've always sometimes sometimes it's not convenient to have. Sometimes I do not have a ruler. So what you can do is when in my research, I actually brought a small ruler. Okay, small ruler. Then plastic tu, normal small ruler. You put beside the fish. I had to take measurement of 400 fish. Right? So it is almost impossible to do it without stressing the fish. So what I would do is I would use a camera. Alright, so a camera that is view from the top. Alright, so I have a white screen on the floor with a ruler standby. So whenever I want to take, after I've taken the weight, alright, so I've laid the fish down on this white screen and then take pictures with the ruler in the picture, right? And then keep that picture later. So I will take my measurement using a software. So I'll do that later, so that the time that you take to measure the fish is very minimal. So you would reduce the handling and therefore you reduce the stress with fish. So that was what I did. So I took pictures and I analyzed the pictures later to take the measurement and the weight. So there are multiple apps now that you can just, I think it works with your phone camera. So just take a picture with some known measurement, then they would be able to, tell the length. If you do not have a big or long ruler like that in the picture. Okay. So this is another thing of IoT, Internet of Things. I think I've mentioned this before. They put in a camera and a probe in the water, and basically it would tell you the size of the fish. Yeah. So basically, I've been using IoT as a tool for my PhD. But for my PhD, actually, I had to monitor growth of fish for 6 months. So I inserted microchip in the fish, right? And then take measurements like that, take pictures, and then analyze the length later using my computer. And then after 6, 8 months, maybe 10 months, right? So I do it again. So I use microchip reader to collect the fish back, and then use take the measurements, morphology measurements. Okay, it is very important for fish farmers to observe their fish daily. Okay, so sometimes they do not do this, alright, but usually farmers, because they want to feed the fish later on in the morning or in the afternoon, so they will be able to tell if something is wrong with it, right? And also to test the water regularly, and also sometimes diseases come not from the water. It comes from those, like I say, if you do not lime your pond, those seafood, right? Those snails, small snails, will come in and bring parasites and diseases. And also think about birds, migrating birds. So migrating birds, they come to your ponds to eat either snails that are near your ponds or your fish. Okay, so some fish farmers have got like coverings on top of their ponds to prevent not only rain but also migrating birds lah. Alright. So sometimes they also have guards around not a person lah, but like a physical barrier on, so they would be able to prevent birds, but also other animals coming into the ponds. Alright, why daily observation is important? Because it allows for early detection, therefore early intervention and diagnosis of diseases. So if there is any problem, you will be able to detect it at an earlier stage, so that you can treat or decide what you want to do earlier, right? So it prevents losses. Okay, so if it is infectious diseases, so you might think about chemotherapy or change depends, right? Non-infectious or infectious or non-infectious, all right? Certainly there would be changes in husbandry, or sometimes we need to use chemotherapy, right? So we will talk about chemotherapy later on other lectures. All right. Certain farms not only want to have grow-out stages, but they also want to breed their fish. Okay. Breeding and genetics are my thing, so maybe I will talk more. So sorry if you do not but anyway. Alright, so when we talk about breeding of fish, we need to think about identification of broodstock. Alright, which males and females that you want to choose to be your parent stock or your broodstock. Okay. So in fish, unlike in cattle, there is very subtle differences between the male and the female morphology. Well, depends on the species. If you're talking about tilapia and grouper, very difficult to determine or to identify unless you examine the genital papilla. And this is not easy, right? Only the experts can know. If you ask me, I will get it wrong sometimes. It's not easy, very hard. Sometimes, people say look at the body depth or the facial or the external, physically external of the fish. Right, so sometimes the male may have a slender body, the female may have a wider or thicker body, but it is not true all the time. Okay, and then when we talk about how are we going to breed, we either choose naturally or artificially. Right, so if it's naturally or artificially, we would use gonadotrophic hormone. Mentioned this before. Somebody asked me in the email what type of hormones. Usually we use HCG, human chorionic hormone something. Right? The reason why we use that is because it is very easy to get. Right, so farmers can calculate it and just inject it themselves. Right, they do not need a vet to be there. Right, usually farmers they do this on their own unless there's a problem, a very very difficult problem, then they will call the vet. Alright, for tilapia, male or female, hard to tell unless you look at the genital papilla, right? So you can see here, for the male, it would look like this, right? So the urogenital aperture is more prominent, right, compared to female, right? So female has a urinary pore instead and an oviduct, but not really easy as this diagram. Sometimes it takes some practice, right? I was supposed to show you another picture. Alright, so if you're using natural, then some species they will be able to breed naturally if you do not inject them. But if you want to, the farmers want to, they are really good broodstocks. They want to manage the time, of when they are brooding and they're spawning and all that. So the females are given one chemical injection to induce spawning, and then they are placed together in the pond. Or if it is an induced spawning, both are induced spawning. Okay, both are induced spawning. It's either that they spawn naturally in the pond, or you want to strip the female and take the male and then mix the sperm and the egg externally. And then you put in it in a tank or whatever. In the tank, you are born in the tank. And once they hatch, then you transfer into a pool. Right? I talked about this before in grouper aquaculture kan. So this is okay. Breeding programs in aquaculture usually when you think about when you think about the number of eggs and sperm that are produced by fish, it is high compared to terrestrial animals. Okay, so they are of high fecundity, but they produce a lot. So fertilization occurs in water externally, so it is something that you can control. So in the previous slide I've shown you, you can do it either way. You want to do it visually in the pond, or you want to induce them. When you want to do it, you can control it. And you can control which female and which male breed spawn, because you can collect eggs from whichever females that you want, keep it, and then choose whichever males that you want, and then make the eggs and sperm for fertilization, right? So a lot of things that you can control in fish breeding. Okay, and the number one trait that we always want in fish, right? And when we talk about aquaculture, okay, not about ornamental fish. Ornamental fish, maybe you would want certain color, certain pattern, but when we talk about food fish, right? So it is always about growth rate, because we're talking about money here, right? So it has an economic importance. Very important is a growth rate. And if you want to be able to control which females and males mate, you need to have a good record of the potential candidates. Okay? So in my PhD, we found out that from our groupers in that specific area that we've looked into inbred. Alright, so there are a lot of numbers, but I do not want you to see all the numbers. What is important is that when we do mass selection, we generally just choose based on the how much eggs or how volume of the milk that is produced. You're only looking at the phenotypical selection only, right? So maybe you think that these males and females are large, they are able to grow very fast compared to other species, other tank mates. So suppose they are the biggest, suppose they are the best, right? But no, not necessarily. These are only phenotypical, selection only. So you're looking only at the phenotypes. You're not looking at what is happening at the genetic level, the genotypes. So if you only select these types of fish, okay, and only in the same cohort, in the same population, and you reuse your broodstock again and again and again, you do not add new parent stock into your farm, alright, it will lead to inbreeding depression, right? So basically, inbreeding depression means if you use the same parent stock again, ultimately those not so good alleles, right, cause poor performance, such as poor growth, alright? So just an example here, I found this paper, okay? Not only does it affect inbreeding depression, affects growth, but it will also affect disease susceptibility. So you think about over a long period of time, although it is easy for you to use the same broodstock again and again, and you think that the farmers would save money if they only use the same broodstock from the same area. You will think that over long term it will cause inbreeding depression, and fish will be susceptible to diseases. So in this paper, so it's the effect of inbreeding on disease susceptibility. So this is Gapisla Gyrodactylus, a parasite. Okay, Prof. Sharif will teach you about parasites. So just read here, I'll give you a tip, right? When you want to do your FYP, if you need to screen for so much paper, right? You have a lot of papers you need to read. Just read the abstract. Okay? Just read the highlight. That's what I do. If I want to think whether I want to use this paper for my research or not, just read the highlights. It's very convenient. Alright, so it says inbreeding effects on resistance to tested inbred individuals had high parasite intensity, less able to clear the infection, and not only that, this is very funny. Parasite infections may raise the extinction risk of inbred population. This is not good. Somehow we still need the inbreds, the inbreds population. But I do not know. You think about it, right? This last statement, is it good or bad? Alright, so there's a graph that they show here in the paper. So it says, alright, you look at this dotted line. Dotted line are the inbreds, right? This red arrow. So inbred fish have more number of parasites, and the parasites persist over longer duration on them. Alright, so that tells you that inbreeding depression is not so good. Alright, so when they have a lot of parasites on their body, and if this is a food fish, the growth performance would reduce, and you would have to fork out money for treatment, fork out money for longer duration of the fish in your farm. And if the fish has got marks, alright, so hemorrhagic or whatever lesion on the body, it would reduce the profit that you will get, okay, because the fish does not look good. People do not want to buy fish that look good, right? Alright, so inbreeding depression leads to the expression of deleterious alleles. Alright, if you have can read this, it's very self-explanatory. So at the very end, if you have inbreeding, right? So these are siblings, right? These are siblings. So and then you mate the offspring of inbred, then you would definitely get the expression of your deleterious alleles. Okay. All right. Selective breeding, if you want to compare cattle, alright, a terrestrial animal, aquaculture, wow, we are lagging, at least 10 years or 15 years behind in terms of technology and the genes that have been discovered, alright. So genetic variation present in desirable traits within a population, right? So you want to select fish not only based on phenotypical traits, but also based on desirable traits in the genotypes. All right, so you want to improve production quality, efficiency, and sustainability. Just an example, right? If you can find those fish which are phenotypically good, and you can test your fish genetically with the sequencing method, then you would be able to save those fish and use them as a broodstock. Right? So the others that are not so good, have a low quality traits, you call them. Alright? So you want the best in your farm, right? The best. Successful breeding programs depends on three things. Alright, heritabilities must be known. Usually these are only known for very commercial fish species such as salmon, trout, tilapia. Alright, phenotypic information needs to correlate with genotypic information. Alright, how do we do that? So we have this so-called superior fish that looks nice or they grow better. So you take their DNA and then you test them using genomic tools. Right. So this is just heritability estimate. So growth is the most heritable, but nothing is above 100%. So the limit here is around 50 to 60%. time. So growth is at the top, followed by morphology, right? Sexual maturity, disease resistance, carcass quality, and end. So these are the traits that are very hot right now. Scientists are really working on looking at all these traits because we can control growth and disease resistance in a farm. Alright, so with introduction, I did next generation sequencing in fish. Alright, so it goes back, selective breeding goes back in 1990 with brook trout. Alright, so trout looks like salmon, but a bit of difference, alright, against parenchylosis. So we have applied high throughput sequencing in fish back in 1980s. But basically, it is to extract genetic information from the DNA. And the reason why we want to do this is so that we can select fish which have superior traits or superior candidates for breeding. So I mentioned before, selective breeding takes years. Alright, so you look at this Atlantic salmon. It took 40 years for them to, produce important results. Okay, high sophistication, and it needs large and highly specialized companies. Right, they look at all the traits that I've mentioned before, and they are able to enhance production by manipulating genes with disease resistance, feed efficiency, and adaptation to movement. So very complex. Usually sequencing needs a facility that is complete, not cheap either. So that's why we need to work with specialized companies to improve this. Alright. Alright, I'm not going to talk about this much because I do not think I have enough time. Alright. So just an introduction what I did for my PhD. We looked at genetic markers called single nucleotide polymorphism, SNPs, and microsatellites. Alright, so in terms of grouper, we have all these. So orange spotted grouper have been discovered, and also brown marble grouper lah if in my studies. So SNP and microsatellite. You guys can read what differences. I'm not going to tell you, not going to explain, but basically these are genetic tools lah, right? So these are kind of like if one fish, alright, look here, if one fish has a difference at nucleotide A and the other fish are G, but A is a better candidate, then you can tell by looking at region A, region 1, alright? So basically that's it. Pretty complicated, but yeah, important if you want to talk about selective breeding in fish. Besides that, selective breeding, okay, important, but also in terms of the quality or the health of the host, which is the fish, we talk about feed and feeding. Okay, I've mentioned before, depending on the species and developmental stage, it's different. And then you want to think about, you want to use natural foods or supplementary feeds. Alright, depends on the cost and the ability of you to culture these natural foods, okay, phytoplankton or zooplankton. Fish food and types. Alright, we have different sizes and different moisture level, and also it can be a floating or sinking pellet. Right? These are just examples. So you can read this types, either dry or non-dry. So what are the moisture level? If it is a non-extruded, extruded or non-extruded is a method of producing the feed. Right? It involves certain techniques. Right? So it can be in paste, cakes or balls, or if it is extruded, is it floating? Those type of things. But basically, what are the things to consider? Firstly, the feed ingredients. Okay, I've talked about this. Do you want to use fish meal? Do you want to use what type of fish meal, right? So you can have a good quality one. You can have a poor quality fish feed meal. It involves cost. You want to include plant formulation products into your formulation, right? Either soy or wheat or certain things like that. And then the feed manufacturing, the type of formulated feeds, right, moisture sink fruit, and it needs to be able to adapt to your feed practices. Okay? The life stages that you are culturing, your culture system, like pond or outdoor cages or whatever it is, right? And also the availability of natural foods. If it's so, you need to think about the schedule which fits your fish species and also your farm management, also the frequency. So these things need to be considered. Not just considered but calculated. Okay. I'm not going to show you how to calculate because I'm not a nutritionist. So perhaps a nutritionist would have better explain how to calculate all this with all the nutritional aspects in mind. All right. We're almost done. How are we? All right, we're almost done. Harvesting. All right. When we have cultured our fish up to a certain period of time that has reached a market weight. Market weight depend right on the fish species and also production or your management. Okay, certain farms provide for bigger size market. Certain farms, want to have a smaller size for whatever reason. Alright. Firstly, for harvesting, it needs to be done days before. Okay, so you need to drain the water partially if you're talking about pond. So not drain all. The fish will die. Drain partially, and then for cages, you raise the cages partially lah. Okay. And then do not feed for 1 to 2 days so that if you're talking about pond, it will reduce water excretion. Itulah when you want to catch them and then put them in a smaller tank or container. So you do not want to pollute that water unless you would have a lot of things to do. You need to change the water there, and that takes a lot of labor. Okay, so purge them for 2 days at least. Also, why you want to feed the fish? They're going to be eaten later on, right? Save your money on feed, right? And stress should be minimal, right? Do not disturb the fish as much, all right, until D-Day, right? Until D-Day. So transfer directly to clean water for transport or storage. Okay. This is very important. If the fish are injured, fetch a lower price lah. Okay, especially if it's a live market. So what they would do, right? They would reduce the water in the pond partially, all right, and then they use nets, right, to gather the fish at one end, and then transfer via either a smaller net, right? And then like for this farm, it's a tilapia farm, so they use basins or buckets to transfer them into the transport tanks. Okay. Transporting live fish, very important. We would want to use lower water temperature. We can do that. We can achieve that by using ice. Okay, why is it important? Use your water temperature because fish are poikilothermic, right? So you can reduce their metabolic activities. That you are going to reduce the stress of the fish. Okay. Usually it comes. You transport them on a lorry lah. So on top has a lot of fiberglass tanks. Those tanks are aerated. Maybe I've shown you a picture last time, right? Those tanks are aerated. And for freshwater species, you would add salt. Alright, why? Because it reduces the stress and salt loss. This is a very favorite question. to be asked in test by my predecessor, right? Why salt is added in freshwater species for transporting live fish? Maybe I would want to ask again. Yeah, maybe, just maybe. Right, very popular question. It has got to do with osmoregulation, alright? When fish, freshwater fish, are in a freshwater, so their gills and their skin need to work hard to maintain the salt in their body. Okay, so when you add salt into their water, so they would be able to maintain homeostasis without losing energy or without using energy, because the mineral transport, or needs energy, right? So you would able to maintain homeostasis and osmoregulation without energy and loss. So read up on that. I'll tell you just basically what it is about. So yeah. Okay, lastly, almost processing and marketing. Okay, because market price depends on the quality of fish. Okay, some fish are sold live. All right, they arrive at the restaurant or whatever it is alive. So they would want to check internally, right? If it is not good, they would fetch lower price. And others like your pangasis, your tilapia might be sold, might come to another processing facility first, right? They're being processed for their meat, for their skin, for bones, whatever it is, or they are being marketed. Okay, so we've talked about challenges in food safety, right? So this is where those knowledge comes in. So you want to think about during transportation and processing, what are the things that could compromise or jeopardize food safety in terms of fish and fish products. Okay, we have discussed that before. Well, this is actually a facility to process fish, but this is a marine fish. Okay, it's not freshwater fish, but more or less it is as intensive as this. All right, so there are rows and rows and rows of people working on the fish, right? Very quick, very efficient. Certainly, with processing plant like this comes not only we're talking about the welfare of the fish, but also the welfare of the workers. There were news back in 3, 4, 5 years ago, but still ongoing, about the concern of shrimp processing plants and the welfare of the people working in shrimp processing. You guys can read about it. But basically, they were saying that the very low cost of processed shrimp and fish translate to the abuse or the abuse, or workers are not being treated properly. They only get very little wage, right, for their work. Those are issues, not only we're talking about welfare of the fish or aquatic animals, but also certainly we cannot escape from talking about the welfare of workers. All right, just an example, you get sushi fish fillet. Okay, certainly my husband always say, I never want to eat tori or pangasis. But certainly you can look up if the management is good, can really know, from tasting the product. Okay, and there's sushi or pangasius, and then there's tilapia fillet. Okay, just an example of what are the final processed fish products that you can get. So I'm going to sum up in a few minutes. So in the previous lecture, we've talked about site selection, what the criteria, water sources, and so on. And then preparation of the pond, drying, liming, transition of water. And then we talk about, okay, so after fertilization, so what will go on in the farm is handling of live fish on the farm, monitoring, pot keeping, fish feeds, transportation, and also processing and marketing. All right, that's it for me. Any questions?
Excuse me, doctor.
Yeah.
I have a question. It's regarding the transportation of live fish, where you mentioned we have to add salt. I was wondering if there is a specific formula or ratio as to how much salt we have to add.
Usually when we talk about salt, okay, salt is a substance that is rarely toxic to the fish. There's a range of dosages but it's not really dosages. For treatment of fish, we would go up to 3 grams or more per liter, right? So for normal maintenance, 10 to 30 grams per liter for treatment. For maintenance or reducing of stress, 2 grams per liter is sufficient. But as I've said before, salt is usually not toxic, so you can work around that range lah, 2 to 5, maybe more. So when we talk about calculation of dosages of chemotherapy in fish, usually non-injectables, alright, things like salt, methylene blue, formalin, antibiotics, whatever it is, we measure the water volume. Right, we do not measure the weight of the fish, we measure the water volume in order to determine the dosage of the drugs or salt that we're going to use. Alright, so that is the range I'm telling you, 2 to 5 grams per liter. We will talk about that. Actually, I will give you a chart later when the lecture comes of the things and the, right? Hope that answers your question.
Yes, thank you, doctor.
Right. Any other question? Alright. Dekat Padlet, did everybody upload their poster? Because yesterday I saw only 14 or 15, so another 4 or 5. So I will check again. And well, the reason I want you to be able to see the work of your friends, alright, so we can share the information and the knowledge. Farha asked me about test 1. I did not receive anything yet, but when I do, I will inform the date definitely. Usually what they did was, so we have lectures for 2 hours. So 1 hour is lecture or whatever it is, whichever comes first. So 1 hour test, 1 hour lecture, something like that. Okay, if there are no more questions, I want to wish those who are celebrating Happy Deepavali, and the rest, and everybody, just please stay safe if you are travelling. Be careful, be extra careful. Kalau orang tua cakap, this is my mother lah, so jangan seronok-seronok sangat, jangan seronok-seronok sangat. Take care of yourselves. Yeah, so I want to say. Alright, thank you. I will end the class. Bye-bye.
Bye, doctor.
Bye.
Thank you, doctor.
Alright.
Thank you, doctor.